last class we discussed about structure and function of plasma membrane. Now we are going to discuss another concept, cell wall. As we have seen earlier and compared the differences in between plant cell as well as animal cell, we have seen the cell wall is present only in some cases, but it is not present in the case of animals. That's what we have to remember. As we have seen, the cell wall was discovered by Robert Hooke. Who discovered the cell wall? Robert Hooke in the year of 1665, and who observed these cells in corpus cell of a plant. He observed in corpus cell. As we have seen, this cell wall is additional protective membrane surrounds the plasma membrane found in plants, bacteria and fungi. In this way, in these kind of animals we can find, in kinds of organisms like plants, bacteria and fungi, we can find the cell wall. How is the nature of the cell wall? The cell wall, it is thick. How is the cell wall? Thick, rigid, very strong, and which is made up of non-living cells and permeable membrane and found around the out of the plasma membrane. So the cell wall, it is thick, rigid, non-living, and it is permeable membrane found out of the plasma membrane. And if you observe the cell wall in different organisms, it is made up of different substances. So the cell wall of different organisms is made up of different substances. For example, the cell wall of the plant, it is made up of cellulose sugars. Plant cell wall is made up of cellulose sugars, whereas in case of bacteria and blue-green algae, that is cyanobacteria, it is made up of peptidoglycan. It is made up of peptidoglycan. Whereas the fungi is also made up of another hard protective membrane called chitin composition cell wall. So, in different organisms, all organisms like plants, bacteria as well as fungi, all are made up of cell wall. But this cell wall composition, the chemical substances of the cell wall is different from one organism to another organism. For example, if you observe in plant cell, it is made up of cellulose. Whereas in case of bacteria as well as blue-green algae, it is made up of peptidoglycan. Whereas in case of fungi, it is made up of chitin. In this way, we can see that the cell wall composition is different in one organism to another organism. If you observe this cell wall, it is a protective membrane. The cell wall is a protective membrane which protects the cell from external conditions. When it is very hot or cold, or when it is rainy or windy, any of these changes occurs in the environment. In certain conditions, the plants cannot move from place to another place like animals, but because they are fixed to a place. So due to this condition, these plants should get protection by themselves. So this protection is made by the plants naturally in the form of cell wall. That's what it is called as protective membrane of cell. additional protective membrane of the cell. If you observe in case of the plant, this cell wall composition is different from one tissue to another tissue. What we are saying, in case of plant cells, why? Because the plant cells are made of different types of cells which are differentiated to carry out their function. So depends upon this variation in their function, the composition of cell wall is different from one tissue to another tissue. For example, if you observe the tissues of plants like the tissues, cells like growing cells, photosynthetic cells, as well as storage cells, as well as parenchymatic cells, and meristematic cells. These different cells of the plants, they were made up of primary cell wall. This primary cell wall of these different types of cells like growing cells of the plant, photosynthetic cells, storage cells, parenchymatic cells, as well as meristematic cells. In these different types of cells, the cell wall is called as primary cell wall. What do we call as primary cell wall? This primary cell wall is made up of cellulose sugar. The primary cell wall made up of cellulose. And if you observe this primary cell wall, the primary cell wall, it is porous and allows the movement of the substances through it. That's what the primary cell wall is porous and permeable. It means allows to move the substances. Permeable. It is primary cell wall is porous, so it is permeable the substances. That is the one function, and it gives protection to the protoplasm. It gives protection to the protoplasm. So protoplasm is nothing but cytoplasm as well as nuclear membrane. These two are protected by means of the primary cell wall. That is an important concept called primary cell wall, which is found in growing cells, photosynthetic cells, storage cells, parenchymatic cells, as well as meristematic cells. These cells are made of primary cell wall, and this cell wall is composed of cellulose. And if you observe this primary cell wall, it is porous and permeable the substances, and it gives additional protection to the protoplasm. And if you observe the primary cells of adjacent cells, so side by side cells, they were connected together by the middle lamella. Adjacent are connected by a cementing material called what it is called? Middle lamella. This is what a primary cell wall found in different tissues. Whereas in case of the dead cells present in xylems, cork, as well as cells, as well as Cork cells, as well as sclerenchymatic cells. They are composed of made of secondary cell wall. They are made up of secondary cell wall. That is the thing. And this secondary cell wall composed of or made of the chemical substances like suberin and lignin. They are composed of chemicals called as suberin as well as lignin. These suberin as well as lignin, they are non-porous, and due to the non-porous in nature, they are impermeable, these substances. So, secondary cell wall is non-porous. It is non-porous, and so it is impermeable. It is impermeable. This is what we can see that another important concept of the fundamental unit of life is cell wall. This cell wall was discovered by Robert Hooke in 1665 in corpuscles and found in plants, bacteria as well as fungi. And if you observe, it is thick, rigid, non-living and permeable membrane. And these plant cells as well as bacteria cells as well as fungi cells, even though they are consisting of cell wall, but their composition is different. The plant cell is made up of cellulose, the bacteria is made up of peptidoglycan, and the fungi is made up of chitin. And if you observe, this cell wall composition is different in different tissues of the plant cell. Like in growing cells, photosynthetic cells, parenchymatic cells, storage cells, as well as meristematic cells. These all cells are made up of primary cell wall. What it is called? Primary cell wall. This primary cell wall is made up of cellulose. And two adjacent cells of this primary cell wall, they are connected or joined by a cementing material called as what do we call as middle lamella. And these primary cell wall cells are porous, so they allows the movement of substances. So it is called as porous, that is permeable membrane. And whereas another important function is it gives the protection to the protoplasm. That is what a primary cell wall. And some other tissues of the plants like xylem, quark, as well as sclerenchymatic cells of the plants, they are composed of different chemicals like suberin as well as lignin, and this cell wall is called as secondary cell wall. It is non-porous and impermeable the substances. In this way we can see that important function of the cell wall and how the cell wall is protecting to the cell. That is we have to see. So if you take a cell and immerse in hypertonic solution. So this is a cell, a container, a beaker. In that we have placed a plant cell. This is the plant cell and outside it is the cell wall. So inside it is plasma membrane. Now a cell is placed in hypertonic solution. What is hypertonic solution means? The concentration outside water concentration is less, inside is more. So now what is happening here in this condition? Due to the hypertonic condition of the solution, it exerts pressure on the cell wall. The solution creates pressure on the cell wall to enter inside. So at the same time, the cell wall is producing against pressure to the turgor pressure or the solution creating pressure on the cell wall. So against pressure is acted by the cell wall. when the solution is creating pressure on the cell wall, the cell wall is creating pressure against the solution. So due to the variation in concentration, so what is happening here? The content of the cell, that is the plasma membrane as well as cytoplasm, they start shrinking away from the cell wall. So the plasma membrane shrinks as well as the cytoplasm shrinks away from it. That is, shrinking means what? They are moving away from the cell wall. Due to the outside pressure, now the water start moving out of the cell wall. Due to this movement of the water away from the cell, that leads to shrinkage of the plasma membrane as well as the cytoplasm. So the shrinkage of cytoplasm and plasma membrane away from the plasma membrane or cell wall, away from the cell wall is called as plasmolysis. What it is called as plasmolysis. So what is plasmolysis means? When the water is moved away from the higher concentration to the lower concentration, that is from inside to the outside of the cell. Due to the movement of the water from inside to the outside of the cell, the plasma membrane as well as cytoplasm, they moves away or shrinks away from the cell wall, and that leads to constriction of plasma membrane. That the shrinking or moving away of plasma membrane from the cell wall is called as plasmolysis. So here, cell do not get any damage due to the pressure of the hydrostatic pressure or turgor pressure of the water. But what is happening here? The cell wall is creating pressure against the liquid. So that's what the cell wall can withstand at higher pressure. That's what the cell wall is, another protection, additional protection membrane of the plant which makes them to protect themselves. That's what the plasmolysis is, nothing but shrinkage of cytoplasm and plasma membrane away from the cell wall is called as plasmolysis. So what is plasmolysis? Shrinking of plasma membrane away from the cell wall is called plasmolysis. In this way, we can see another important concept is plasmolysis. This is because the hypertonic solution which is exerted pressure on the cell wall, that leads to the movement of water from inside to the outside, and that leads to the shrinking of plasma membrane away from the cell wall, and the process what we call as plasmolysis. In this way, we have seen another important concept is cell wall and types of cell walls in different tissues of the plants and plasmolysis.